Another important technique that can be used in gene therapy is gene silencing, which is basically turning off a gene so that no protein is produced from it. Triple helix forming oligonucleotide gene therapy targets the DNA sequence of a mutated gene and prevents its transcription. The procedure involves delivering oligonucleotides, which are short and single-stranded pieces of DNA that bind to the groove between a gene's two DNA strands. This binding results in a triple helix structure, meaning that it cannot be transcribed into mRNA.
Another form of gene silencing is called ribozyme gene therapy. This targets the mRNA that's transcribed from the gene. Ribozymes, not to be confused with ribosomes, are, stay with me here, RNA molecules that act as enzymes that act as molecular scissors that cut RNA. In this procedure, ribozymes are designed to find and destroy the mRNA transcribed from the mutated gene so that it can't form proteins.
The last form of gene therapy that we will be talking about is a procedure that involves genetically modifying immune cells to target and kill specific molecules. This procedure involves isolating an individual's immune cells and engineering them to recognize a certain antigen. An antigen is a particular molecule, such as protein on the surface of a cancer cell. Upon returning to the human body, the modified cells will destroy any and all cells that carry this specific antigen.
You might be asking yourself, why is this so important? If technology is improved, changing one's physical or mental health could be as easy as getting a flu shot. There are many positive aspects of gene therapy. It can wipe out genetic diseases before they can even start to spread, if they're caught early enough, and they can eliminate suffering for future generations. Gene therapy is also a surefire technique for diseases that haven't been researched yet. This is because everyone carries defective genes, whether they're aware of it or not. Gene therapy can control or eliminate hereditary diseases. As for now, the cost of gene therapy is very high. However, as the technology grows and becomes more accessible, gene therapy will become cheaper, especially because usually it's only a one-time procedure. Scientists are trying to pursue this technology because if it's mastered, the act of finding a faulty gene and replacing it with a working gene will become seamless, which is a big step for humanity.
Through the rapid development of rDNA technology, the overall understanding of the genetic basis of a human disease has facilitated the development of gene therapy. Gene therapy involves complex cell processing, which can be time-consuming and expensive. More simplifications are necessary in order to determine the suitable usage of gene therapy.